It's a real pleasure to welcome Ilkka Hanski to give a Storer lecture. There's a long description of Ilkka's accomplishments in your program, and I'll just mention one or two highlights, and there's one or two things that are not in here. Ilkka received the Crawford Prize in Biosciences in 2011, which is a huge honor. He's a foreign member of the Royal Society in the United Kingdom and also a foreign member of the US National Academy of Sciences. He received his PhD doctoral degree, it's not a PhD, from the University of Oxford. He is currently a Finnish Academy professor in Helsinki. And the one thing that's not really mentioned is one bit of expertise is that Ilkka's some of his first important work, and he's a real expert on dung beetles, which somehow I tried to get into this program, and the person who wrote it refused to put it in. He'll be giving a talk tomorrow at 4:10 in Geet Hall, so you don't want to miss that. That's on eco-evolutionary spatial dynamics. And today he'll be giving us a talk on environmental biodiversity, human microbiota, and allergy. Please join me in welcoming Ilkka Hanski.
Thank you very much, Alan, for flattering but accurate introduction, including the dung beetle part. Now, it's a real pleasure to be back in Davis. Alan remembers, and Susan Harrison remembers, perhaps nobody else knows that, that I spent actually half a year in Davis 20 years ago, and I was here 6 years ago as well, so I really enjoy coming back here. Now, my presentation today is about what you might call two global megatrends. The first one is real familiar to all of you, and this trend is the declining biodiversity locally, nationally, and globally. The three lines here are the WWF Living Planet Index, which summarizes trends in populations and species of all kinds globally. The two other lines are for bird populations, also globally. And as we all know, there is this real dramatic decline in biodiversity all over the world, such that the Living Planet Index has declined by 30% or so in 30-40 years. The other megatrend that I would like to discuss today is just the opposite, dramatically increasing trend in the incidence of asthma, hay fever, like here, allergies, other kind of allergies, but also in various other inflammatory diseases, which include type 1 diabetes, some cancers, some people say even depression has an inflammatory basis. Now, these data come from Finland, but I'm pretty sure that the US data would look pretty much the same. Now, 2 years ago, just by chance, actually, I happened to start talking with two medical doctors who are working on allergies, Lena von Hertzen and Tari Hahtela. I know Tari because Tari is very keen on photographing butterflies, and I like butterflies myself, so it's our common point. And we happened to start talking about these two megatrends, and we sort of suddenly came to the idea that maybe they have something to do with each other. And actually, Tari particularly, he had been thinking along these lines for some time before. And in a nutshell, the way they might have something to do with each other is because there's this interaction or likely interaction between the three hugely complex systems of natural biodiversity, including the environmental microbiota. The microbiota which we have on and in our bodies, which again is hugely diverse, has thousands of species, my body has 10 times bacterial cells compared with my so-called own cells, and so on. And thirdly, we have the immune system, which is another hugely complex system, not with lots of species, but with lots of molecules which are interacting with each other. Now, as I understand it, and of course I'm not an immunologist, so if there are any immunologists in the audience, I already apologize for probably using some naive terminology here. But anyway, I understand that there's a pretty well established understanding of the interaction between the commensal microbiota and our immune system. The microbes, they stimulate various cells which are part of the immune system, which either have pro-inflammatory function, or they relate to that sort of response, or anti-inflammatory function. Now, it's also very likely, even though I guess the data here is much less conclusive, that our commensal microbiota is interacting perhaps one way, but nonetheless interacting with the environmental microbiota, such that whatever environmental microbiota we are encountering, it will influence the composition of our own microbiota. Now, if that sort of normal, and of course I should mention that during the evolution, of course, we have been evolving with the microbiota all the time, and so in that perspective, it's quite likely that such interactions would be important. Now, if that natural state of affairs would be somehow disturbed, perturbed, one example being you taking antibiotics, you will mess up the flora in your gut. Now, that most likely will have some consequences for the interaction between the commensal microbiota and the immune system. But also, according to our hypothesis, another way of messing up with the commensal microbiota would be to reduce the contact between you and your microbiota and the environmental microbiota, and in a broader sense the natural environmental features in your surrounding, which might then lead to a situation where your immunological tolerance is not functioning anymore in the way it used to function, it should be functioning. Now, this idea is not really fundamentally that surprising. It's related to various other ideas in this field. I'm sure that most of you have heard about this hygiene hypothesis and the notion that, which is pretty well established, particularly in Central Europe, that growing up on a traditional farm with cattle is about the healthiest environment for you as a small child to grow up. This will lead to a well-developed immunological tolerance. You are unlikely to develop allergic sensitization and such. So what we are now proposing basically is to sort of take the hygiene hypothesis out of doors. So not only the conditions inside your home, but also in the broader environment might be involved. And of course, the idea is also related to the understanding that we now had, that the microbes are playing a real important role in the development and maintenance of the immunological tolerance. So the idea is all right. So what we have now done is to conduct an actual case study to test the idea. And the case study is based on a random sample of school children living in eastern Finland, within a region which includes a small town, includes villages, but also isolated farmhouses in the rural area. And this really is sort of critical for what we have done. The children are living in a heterogeneous environment where there is variation in the type of surrounding environment in which they inhabit. I don't have a map of Europe, but this is in Europe. This is Russia. This is Finland. And so what we are doing here is taking 4 pieces of data, 4 pieces of evidence, and we want to explore how are these pieces related to each other. The first piece relates to environmental biodiversity, which we have measured at 2 levels, as I will explain. We have characterized the commensal microbiota by taking a DNA swab from the forearm, and then using the metagenomics methods, we have characterized the bacterial composition in this sample, so the skin microbiota. We have measured the immunoglobulin E antibody levels in the children to characterize their atopic sensitization or allergic disposition. And finally, we have done some measurements of the immunological response in the same study subjects. So the question really is, how are these components, how are these pieces related to each other? Well, so the first piece is really to just characterize whether these individuals are healthy or atopic. And as I already mentioned, this is done based on the measurement of the immunoglobulin E antibody levels in blood samples. Now, quite conveniently, the distribution happens to be by model, and so we are characterizing this first mode as the healthy, or they constitutes of the healthy individuals, and these are atopic individuals. There's a little peak in the middle, which is a bit annoying. To make sure that it doesn't really change the results, we tried to place the cutoff point in a somewhat different location, and it didn't really affect the associations of atopy with the variables that we were looking at. All right, so the second piece of evidence then relates to the skin microbiota, and as people do in this context, we use the 16S ribosomal gene to barcode the bacteria in the sample. Now, in the same way as previous studies which looked at the skin microbiota, there were these 6 classes of bacteria which were dominant. This column here gives the relative abundance of the 6 classes, and this column here gives the numbers of genera. It's not possible really go to the species level, so we stayed at the genus level. The total number of bacterial genera in the pool sample was 572. So it's a very diverse community. Well, the problem here is that there are so many variables, 572 bacterial genera, so most of them are really rare. I summarized the variation in the generic diversity in the different bacterial classes by doing a principal component analysis, which, as I said, summarizes the pattern of variation in the data. The first principal component typically is correlated in this kind of material with all the classes. It just reflects the fact that some samples happen to have more of bacteria of any kind than other samples, partly for technical reasons, partly for possibly also for intrinsic reasons. Some people just have more bacteria on their skin than others. The second principal component, as you can see here, it is positively correlated with all proteobacterial classes and negatively correlated with all the other bacterial classes. And I'll get back to that in a minute. The third piece then is the environmental biodiversity, and we measured that at 2 different spatial scales. The broader scale was a circle with a radius of 3 kilometers around the home of each of the study subjects. And here we just calculated the 5 or the percentage coverage of the 5 main land use types in the study area. We are in eastern Finland, so there's lots of managed forests. There's quite a bit of agricultural land. There are built areas. Some of the youngsters, they are living in the single town which is there, and then in villages there are also lots of lakes, so water bodies, and there are some wetlands. So these are the average coverages in the pooled data. And again, I wanted to summarize it with one or two variables, and I did a principal component analysis of the land use types. And as you can see, the first principal component in this case, it is positively correlated with forest and agricultural land, and negatively correlated with built areas and lakes and water bodies. So it kind of reflects the amount of vegetated habitats in the surrounding. Here's the first result. We are now looking at the relationship between the first principal component, which I just described to you, of the land use types, and the second principal component of the generic composition of the bacterial classes. And I think on the next slide I have a reminder that on the land use principal component, this end of the axis we have lots of forests and agricultural land, so these vegetated habitats. At this end we have built areas and water bodies, and on the bacterial axis here we have high diversity, generic diversity of proteobacteria, here more of the other bacterial classes. The relationship is not very strong, there is a lot of scatter, but it's still highly significant, indicating that the children which have lots of forest and agricultural land in the surrounding of their home, they tend to have lots of proteobacteria on their skin, and then vice versa for the other bacterial classes. So the first conclusion then is that the land use types have an effect on the composition of the skin microbiota. And surely the causality goes that way. It's difficult to imagine that the skin microbiota would affect the land use types. Also, if we look at the health status of the individuals, dividing them now in healthy and atopic individuals based on the immunoglobulin E antibody levels, there is a quite dramatic effect of the land use types as characterized by the first principal component axis, such that the individuals which are living in dwellings surrounded by more forest and agricultural land, they tend to be healthy, whereas for the atopic individuals, there are more atopic individuals if the home is surrounded by built areas and water bodies. And this is highly, highly significant effect. So we have this arrow pointing from land use types in the surrounding of your home to the incidence of atopy. Next, we went down to the yard level. We characterized the biodiversity at the level of yard surrounding the home. Now the homes are really different, including, some people are living in flats, in apartment buildings in the town, or even in larger villages. Some people are living in twin houses or row houses. Some people are living in isolated houses. Regardless of the type of the dwelling, I hired 10 undergraduate students to visit all these places to describe the cover types of the yard, but also identify all the vascular plants in the yards. The average was 150. So average yard in eastern Finland has 150 vascular plants. So we look at then possible associations between the numbers of vascular plants in these 5 different categories, also dividing the plants into common, sort of dominant species, and uncommon species with only single individuals or scattered individuals. And there was surprisingly one association that jumped out from these data in one group of plants. The title here is a bit misleading. The title should say species diversity of uncommon native flowering plants. So it doesn't include decorative plants, but native plant species and uncommon ones. The axis here shows a residual where we have taken into account the fact that sadly not all our undergraduate students are equally good in identifying plant species. There was some variation among the pairs of students. We took that into account. The significant effect here would remain even though you don't take that into account, but it's worth taking into account. So this is basically the number of uncommon native flowering plant species on the yard. We are plotting it here against the total number of flowering plants on the yard to kind of take into account the variation in the size and possibly other properties of the yards, which I think is a sensible thing to do. And when you do that, you can see that there's a difference between the yards of healthy people and yards of atopic people. There are more native flowering plants in the yards of healthy individuals than in the yards of atopic individuals. And that's, again, is a highly significant difference. Now, what is the mechanism? We surely don't know, and I'm happy to discuss this after the talk, but the pattern is here. And this variable was uncorrelated with the principal component of the land use type. So both factors, they influence the incidence of atopy in a logistic regression model. Okay, so an arrow pointing from flowering plants as a measure of biodiversity at the yard level to atopy. Let's then move on to the skin microbiota. Remind you that there are these 6 dominant classes. And what I did here was, again, to do a very, very simple association test between atopy and the relative abundance of the 6 main bacterial classes, as well as the generic diversity in the 6 bacterial classes. In the case of the relative abundance, there was nothing. This is a p-value of that association test. In the case of the generic diversity, there was nothing down to this point, as you can see, but then suddenly there is this fantastically small p-value which is jumping out from the data. And it's coming from this pattern. So here we have the number of genera in gamma proteobacteria on the skin of the study subject, plotted against the total number of bacterial genera. Now it's very important to take into account the effect of the total number of bacteria in one way or another. One could use rarefaction, but actually in this case, when the gamma proteobacteria are rare, the rarefaction is less efficient, has less statistical power than by simply correcting with the total number of bacterial genera in the sample, which reflects the biologically meaningful size of the sample. And what you can see here is a really quite, I think, a dramatic difference in the number of gamma proteobacterial genera, which was higher, significantly higher on the skin of healthy individuals than on the skin of atopic individuals. And again, that factor was only weakly correlated with the other factors, and it entered the common regression model. So they all had a significant, they all made a significant contribution. So we have this one particular class of bacteria in the skin microbiota, which has this strong association. There's also a weaker arrow coming from the land use types to gamma proteobacteria. This is not quite as strong an association as in the other cases, but still is statistically significant. Now, in the case of the association between atopy and the generic diversity of gamma proteobacteria, the causality is not quite as clear as in the other cases. It's conceivable that atopy itself, the condition of a person, might somehow influence, might somehow encourage greater diversity of gamma proteobacteria on the skin than in a healthy person. Now, we actually have several lines of several arguments to support the causality from gamma proteobacteria to atopy, but I just discuss with you one of these lines of argument. And that relates to the expression of various cytokines, which are sort of signaling molecules in the immune system. We have measured many of them, but I'm focusing here on this interleukin 10 cytokine, which is, as I understand, not an immunologist myself, but what I've been told is that this is a key anti-inflammatory cytokine in our immune system. So if the blood cells express more of this signaling molecule, that tends to suppress any inflammatory response. So we have measured the expression of this cytokine in peripheral blood mononuclear cells, which however doesn't yet, we don't really know which particular type of cells of the immune system are involved, which would be real useful knowledge, and we are currently working on that. But nonetheless, some cells, well, there may be several types of cells which are expressing this cytokine. We are measuring the total amount of expression. So once again, I did the same sort of very, very basic association analysis by just correlating the expression of this signaling molecule now separately in healthy individuals and in atopic individuals with the relative abundance and separately with the generic diversity of the same 6 bacterial classes. These are again the p-values, and the sign shows the sign of the association. There is one p-value which is nominally significant, and interestingly, it again involves the same class of bacteria which were involved in the previous associations, gamma proteobacteria. And in this case, unlike in the previous cases, the association actually is with relative abundance of gamma proteobacteria, not with the generic diversity. Now, given that it's in the relative abundance, one may ask whether there is some particular genus of gamma proteobacteria which is more strongly than the others associated with the expression of this interleukin 10 cytokine, and that indeed is the case. There is one particular bacterial genus, this unfortunately is misspelled, it should be Acinetobacter, gamma proteobacterial genus, which is amazingly strongly associated with the expression of this interleukin 10 cytokine in healthy individuals, not in atopic individuals. And this graph now shows the association separately in the healthy individuals and in atopic individuals. The p-value here again is something like 0.0009 for the interaction. So in atopic individuals, in the healthy individuals, more the study subjects had this Acinetobacter bacteria on the skin, greater was the total expression of this interleukin 10 cytokine. Whereas in atopic individuals, there was no positive relationship. In fact, there was a slight negative relationship. Now the interpretation here is that more you have this bacteria on your skin, they are stimulating the expression of this cytokine, which has this anti-inflammatory function in healthy individuals. Whereas in atopic individuals, something else is going on, and this association has broken down. Now, another sort of pretty amazing thing is that this analysis led us to identify amongst the 572 bacterial genera this particular genus, Acinetobacter. And it so happens that people who are working with asthma using the mouse model, they have identified a species of Acinetobacter, the same genus, which has a strong protective effect in the mouse model against asthma. So this, I think, is pretty strong evidence for the causality also in our material of the Acinetobacter really having this effect of stimulating the expression of this anti-inflammatory signaling molecule. So I come to the conclusions. To me, the natural interpretation of the data is that more contact we have with environmental biodiversity, more diverse is the commensal microbiota on and in our bodies, and greater is the immunological tolerance. And therefore, we can conclude that these two trends, these two megatrends, the declining biodiversity in the environment and the rapidly increasing prevalence of various inflammatory diseases in developed countries, particularly in urban environment where people have less contact with natural environmental features and biodiversity. Indeed, they may be linked with each other, as I've been explaining. Finally, I want to thank a large number of people. This has been really a fun project because it has involved researchers with very different backgrounds from ecology to allergology to bacteriology, immunology, and microbial metagenomics. Also want to thank a large number of agencies for funding the research. I want to thank you for your attention. Thank you.
Sure, we'll be happy to answer questions.
Yes.
Your medical colleagues have any suggestions for therapies based on this?
Well, I mean, one could start commercializing gamma proteobacteria and rubbing it on your skin, and maybe that will be helpful.
Dirt capsules.
Yeah. Now, this, of course, is widely done in the case of gut microbes, as you know. Like, if you are taking antibiotics, you are also advised to take some probiotics to maintain or restore a healthy gut microflora. Yes.
So I know nothing is known about the natural history of a lot of bacteria. Is anything known about your beneficial bacteria?
Well, if you Google gamma proteobacteria, you will find that it is a very, very diverse, heterogeneous group of bacteria, including many pathogenic species, but most of them are just regular bacteria in the soil. They seem to be quite common on surfaces of plants, which may or may not be important. But I don't really know much more than that. One interesting feature which I'm finding really interesting is that the associations with gamma proteobacteria and atopy was between the generic diversity of this class of bacteria rather than their relative abundance. And that's kind of not perhaps what you would expect. You might expect that more you have this good bacteria in your skin, that's the better. Why is diversity important? Now there are notions that actually the diversity of commensal microbiota can be important. Other studies on asthma and allergies. There's also the possibility that this relates to the history of allergic sensitization in these individuals. We sampled them when they were 15, 16 years old. Now they have become sensitized much earlier in their childhood. And so perhaps it is the case that the diversity of this class of bacteria is a more constant factor in the skin community than the relative abundance. Maybe the relative abundance of one class of bacteria is responding to all kinds of changes in, I mean, you may be washing, or the children may have washed themselves with some particular soaps, and that may have kind of perturbed the relative abundance more than the composition reflected by the generic diversity. Well, I don't know, but that was the pattern. So the diversity itself was was involved in these associations.
Just as a follow-up to that, if you diversity in an agricultural crop can prevent outbreaks of any particular kind. So it may be that having the microbial community without these sort of density compensation bursts that might sensitize one might be important.
Sure. Well, I don't know really.
Well, I'm guessing.
Yes. But these are important guesses. And by the way, I didn't mention that, of course, it would be lovely to also measure the environmental microbiota, to characterize the environmental microbiota, not only the microbiota on your body. But I don't really know what would be the relevant measurement. You go to a forest, and how do you sample the environmental microbiota? You can do it, but would that be a relevant characterization of the environmental bacterial community, which is potentially is influencing the community on our bodies?
Maybe you could send a little kid through the vegetation wearing.
Sure. yes. Well, that might be the optimal sampling.
Yeah.
You were first.
So basically, your study lends big support to the hygiene hypothesis.
Yes, exactly. It is strongly related, and really the main difference is that the hygiene hypothesis, as I understand, originally was conceived as relating to the hygienic conditions in your home, whereas now we are sort of taking it out of doors, and we are considering the microbial diversity, composition, our contact with that in the surrounding broader environment. Yes.
And the diversity on the skin is likely, just seems to me, it is likely to be a proxy for bio contact with microbial diversity with other organs. Right. Is it likely that the skin is determining organ?
Well, I mean, once again, we don't know for sure. You have to sample the microbiota on bodies somehow, and so there basically are three components. There's the skin component, there's the gut component, and then the component in respiratory channels. And the skin is the easiest to sample, and I guess that's the main reason why we decided to sample the skin. But again, it would be important to see whether there is a sort of overall correlation with the other parts of our commensal microbiota in this respect. Yes?
You had some remarkable correlations between the microbiota and the environment of the children and atopic. So I was wondering if you had measured other variables that could also influence microbiota, like diet,
good point.
activity.
I should have mentioned that we did, and indeed this cohort school children has been participating in a long-term study of allergy for 7 years. And in this sample, which is rather small, 118 children, there were no significant correlations with any of these other factors, the ones that you mentioned, and also including things like whether the child has had a pet, or whether a family member is smoking, or the condition of the house, whether it's good or bad. There were no significant correlations. There were associations in the directions that you might expect, and with more statistical power you might demonstrate significant association. What really is, to me, is kind of very, very surprising is that in a rather small sample like that, you can still find these associations with these environmental variables. And I can only speculate on the reasons for that. The reasons may include that these children have been living in the same house, same home, and surrounded by the same environment during all their childhood. So that interaction has been kind of pretty constant. And also the sample is pretty good. These children have been randomly selected amongst the school children in this from this area, and they do live in very different kinds of environments. So there's variation, therefore, and you may pick up these relationships. Yes. Hi, Andy.
Picking up on your point that it wasn't the total amount of these bacteria, but the diversity. So actually, how do you relate this to even sort of classic issue in community ecology of what explains variation in diversity in species, or gene diversity, generic diversity? Is it limited by how much stuff is coming in, and that's what you're thinking, or is there a role here for even different habitat, different sort of environments on their skin that actually affects their diversity? What's controlling diversity there, and how does it relate to the overall picture?
Well, once again, excellent questions, and unfortunately I don't have too many answers. In the first place, the total abundance of bacteria is something that we can't really measure, because depending on how you take the sample, that has a huge influence on that. So what we were looking at was the relative abundance of these different bacterial classes in relation to the sample abundance. But nonetheless, I would have expected that this relative abundance would have been important, and it wasn't, except in the case of this Acinetobacter result. Now, exactly what is the relationship between the and how does this commensal microbiota on and in our bodies, how does it become assembled in the course of our life well, there is some understanding of that. Early childhood is critically important. It's well known that if you are born with a cesarean, you start off with a very impoverished microbiota in your body for obvious reason, and that effect actually has a lasting effect. There are US studies which suggest that this is one, I don't know whether it's major, but anyway, it's one cause of increasing incidence of allergies, actually, more cesarean births. But then in the course of your life, you do accumulate your bacterial community, but exactly how that happens and what is the turnover, and what happens if there is a major perturbation, I don't think anybody knows. And of course, the one reason is that it's only during the past 5 years that with these new sequencing technologies that people have been able to start doing these studies where you can actually relatively easily characterize the full community of bacteria in our body.
It does sound like your overall hypothesis is almost like the beta diversity, or just the diversity in the overall habitat ends up determining what you get exposed to, that determines the diversity on your skin,
right?
As opposed to even things like they compete and they other sort of community ecology ideas about maintenance of diversity.
Well, I'm sure they compete. I mean, surely, there are hundreds of bacterial species on 1 square centimeter of your skin, and there must be strong interactions. There also, by the way, there are suggestions that, like, if you are washing yourself much, you have a higher probability of actually acquiring some pathogenic bacteria which find it easier to colonize you if you have got rid of the beneficial community in the first place. So don't wash too much. Susan.
Curious what we know about the geography of allergies in addition to kind of urban to less developed gradients. Are there other patterns? You think from what you said that allergies should be low in the tropics with tons of diversity everywhere.
Well, I don't, to be honest, I don't know. But what I do know after having interacted with these people is that allergies are rapidly expanding, like in cities in China. It's becoming an equally major problem than in North America and Europe. So there is this urban-rural gradient, and my understanding is that for sure there is a big difference between the developed countries and the developing countries, with the exception that with urbanization in the developing countries, they are catching up. Once again, I want to perhaps emphasize that we were talking about, or we were studying this atopy as an example, but we are not talking about only allergies and asthma. We are potentially talking about all kinds of diseases which have an inflammatory basis. And as I mentioned, this includes type 1 diabetes, many cancers, and also other things. So it potentially is a big thing.
So what about the likelihood of positive feedback loops that through the behavior of the people? So if they have the allergies and they further avoid contact with things that would even actually develop their bacterial diversity that would help them shield off allergy, but they're not going to do that.
Yeah.
The ones that don't have allergy actually just allow themselves more exposure and even helps them stay
with
allergy.
I mean, I think at least in Finland, the treatment for normal or sort of weak allergies is now to try to develop your tolerance and not to sort of try to avoid the contact with the allergens. So my understanding again is that, and I'm sort of speaking the area which I don't really know too much about, but my understanding is that the early childhood is really critical. But it's not only that. It also depends on our lifestyle and potentially with our contacts with the surrounding environment and the hygienic condition in our home during our entire adult life. There's also interesting kind of piece of evidence for that, namely among immigrant populations which are coming from poor countries to developed countries in Europe and I guess in North America. Typically takes about 10 years before these adult individuals acquire many surprising. Some gamma proteobacteria. Yeah. Yes. Okay, that was the point. Yes.
So sort of going along with what he said about it doesn't take that long in Central Valley. And your thing about land cover said that agricultural areas tended to have lower incidence of atopy. But living in Davis, it's sort of famously, people who've never had any problems with hay fever move to Davis and they get allergies because, and the explanation I've usually heard is that there's just so many plants all blooming at once with so much pollen in the air.
Yeah.
But so is it that there is not enough diversity of pollen, or?
Yeah. Yeah, I'm not trying to, well, I would like to propose that, gamma proteobacteria is the only thing which matters, but I guess it's not. And, it's, I'm sure if you are exposed to to masses of particular type of pollen. Like in Finland, perch pollen allergy is the most common type, and here may be something else. Regardless of your immunological tolerance, that massive attack may be too much. I guess that's about the only thing I can say.
One way to step back and talk about what you've done here is you've used ideas from ecology in some sense to look at a essentially medical issue. So that's at least one, sort of what led you to do this to some extent, and two, do you sort of envision any other similar sorts of forays?
Well, as I mentioned, it really was a chance encounter with these medical doctors who have been working with allergies all their life, and the contributing factor was that one of the doctors is keen on butterflies, and so am I, and so it's a chance. What I really like about the project is that it's sort of a grassroots development of this idea. It was not that some agency was telling that ecologists and medical doctors should get together and start working on the allergies, but the idea just came from these individual interactions. And as I mentioned, I found it, and I have found it really exciting that people from very different backgrounds come together and all are about equally excited about this as I myself, including immunologists. Or maybe we have been very lucky and we have encountered a couple of immunologists who can start thinking in these terms, and are happy to interact with ecologists. Yes.
The finding of the really strong association with Acinetobacter, was it?
Yeah, Acinetobacter. Yeah.
It's great that you have this sort of outside evidence to validate that, but you mentioned that we're increasingly able to sequence a lot of bacteria and maybe identify them at some type of level, and as we are able to generate more and more potential explanations of these type of associations, we'd expect that just by chance we could find some really strong signals.
That's right.
And we might not always have that type of mechanistic validation. And so it might be a great way to generate new things to look at. But absent that research program, are there mechanistic ideas that we can use to sort of sift apart the wheat from the chaff? Do we need new statistical techniques like in genome associations, or is there a cautionary side to this tale?
Right. Well, maybe, and I totally avoided mentioning anything about multiple testing and that sort of problem, which is here. So I kind of purposely reduced the number of variables to a very small number to avoid it. But my feeling is that any one of these associations in isolation would not be that interesting. But the assemblage of these associations, which have these common elements like the gamma proteobacteria, with this, as you mentioned, this external evidence coming from these mouse model studies, I find that kind of real encouraging, and certainly suggests to me that there is something real going on here. We are now trying to actually to identify, this Acinetobacter is a genus with again, surprisingly a very diverse genus in terms of what kind of bacteria are there. There are about 20 species which have been described, including Acinetobacter baumannii, which some of you might recognize. It's a pathogenic species. It has, if you Google it, you will find that it has been particularly common amongst US soldiers coming back from Iraq. When they go to hospital, their wounds become infected by Acinetobacter baumannii, which is causing a real problem. So why is it so beneficial, this genus in this case? Now, we are now in the process of trying to figure out exactly which species of Acinetobacter we have here. It's kind of difficult because if you just have this one single gene, information there is not quite enough. You should sequence all of them, and that's a fairly expensive operation for material of this size, but we are sort of working in this direction. And as I mentioned, the other thing we are now doing is to to measure more of the expression of these cytokines, which form a sort of pathway, and there are interesting correlations in the expression level of the different cytokines, which then associate with the, in this case, with the abundance of this Acinetobacter. This is again, maybe it's it takes an ecologist to suggest that you should do that, because if medical doctors would, when they are working on that, they take their blood samples from healthy blood donors. They completely disregard the information, potential information concerning these blood donors, and they just work on these molecules. Whereas here, I think the key is that you actually know something about the people, where the blood is coming from, and then you relay that information to what is happening at the cellular level, which I find reasonable. Yes?
I have to admit, actually, you did not convince me with your theory.
Okay.
And one of the reasons is that when you had your first slide in the Finland, like in the 70s, practically no asthma and all that, and then this kind of gigantic race. So a lot of your children who lived in this kind of nice bucolic landscape of agricultural and forest environment had also a lot of atypic. It was only just slight trends you could show. So I'm sort of wondering whether it's just one minor factor, but the actual real big difference is still somewhere else between 1970 and today.
I'm not sure if I followed you, Agnes. So sorry. Maybe jet lag.
I mean, it's an interesting study, and it has to be done. And my first reaction is it has to be done again and again.
Of course, and one has to see how general the results are.
Yeah, but so you had this one slide which shows that there's almost no allergies
in the 70s.
In the 70s, but your cohort was sort of split between people who lived maybe somewhat like a lot of people lived in the 70s, i.e. in the middle of forest and wide biodiversity landscape. And others were in these more restricted environments, you know, let's say in cities and in departments and so on. Why did have a lot of these kids which lived on the land also pretty bad data?
Well, I mean, there's always variation, isn't there? I mean, it can't be a sort of deterministic process.
Because what would happen is if you could have gone back to 1970, would you have seen the same data? That's the question.
Yes.
And then you don't have the asthma plus or minus. And so my personal feeling is...
Well, I mean, you still would have, but it's only that, the proportion of individuals with this atopic sensitization has rapidly increased in Finland and the US and in lots of other...
Obviously also in the country.
Also in the country, but, of course, we are not saying that, if you are living in a rural area, there's an absolute protection against any kind of inflammatory disease. It's only that this could be a factor which influences your immunological tolerance and the development of your immune system. But, I mean, of course, you are right that what we have presented is just a case study, and I think there are interesting associations. Now it's important to see how general these findings are, and is it possible to find in other populations also comparable associations, and would also the gamma proteobacteria be involved in these other associations? I don't know. Yeah.
So reminder, tomorrow, another talk that will include, I am sure, something about butterflies.
Yes.
And let's thank Ilkka again. So has this inspired you to think?